All right, so a few of you had asked whether or not I could more or less narrate the videos on cellular respiration. So I'll go through these videos that you have posted online, and hopefully it will give you enough information of what you need to know and kind of get rid of all the other stuff. So just to keep in mind, we're talking about cellular respiration, and we know that we have glucose coming into our cells through the process of diffusion, and it's a pretty big molecule, as you notice, with 6 carbons. And if you are a plant, you probably made it in that cell with your chloroplast, so it actually didn't really move into the cell. It was produced in the cell and is being used in the cell. And if you are anything other than an organism that photosynthesizes, you probably consumed it, or you had it diffuse into the cell. So with that said, make sure that this is running here. I'm moving this ahead. The key to cellular respiration, again, is to make as much ATP as possible. And in that glucose, you have these chemical bonds. You have carbons and hydrogens bonded together, and they keep glucose. So the first step is ATP attaches one of its phosphates into each side of the group. So 2 ATPs are used. And with the help of enzymes, that breaks it into 2, which is called glyceraldehyde 3-phosphate, or G3P. All right? And remember, it's enzymes that are doing all of this work. What happens next is that an energy-carrying chemical chemical, NAD, will form to be NADH. So it's taking some of that energy. Here we have ADP becoming ATP. And let's see what happens next here. Always takes a second. And again, enzymes rearrange it. 2 waters are going to leave, 1 from each of those. And an ADP gets made into an ATP. And that is now in pyruvate. All right, so as a result, we've used 2 ATPs, but made 2 NAD into NADHs. So the next cycle that's going to be happening is called the Krebs cycle. And I have, in this case, those 2 pyruvates that were made from glucose that would happen during glycolysis. And as you can tell, they're smaller molecules, and because they're smaller, they're able to fit into the mitochondria. So eventually you're going to see one of these pyruvates. Perhaps it helps for me to press play. So you have these pyruvates slowly entering into the mitochondria, particularly towards the center. And this is where the Krebs cycle happens, again with lots of enzymes doing most of the work. And the key to the Krebs cycle is making NAD into NADH and FAD into FADH2. And remember, it's more or less charging those half batteries, okay? NAD and FAD are half-charged batteries, and they are going to be charged. So we start with the pyruvate changing into an acetyl-CoA. That acetyl-CoA is going to attach to a chemical that's in that mitochondria, just being recycled over and over again. And as a result, you have 2 carbon dioxides leaving. There's one of them leaving. And an NAD getting charged to be an NADH, another carbon dioxide leaving, another NAD being charged to NADH, an ADP being charged to an ATP, and then you notice that you had an FAD being charged to becoming FAD2, and one more NADH getting charged. And you're going to see that cycle one more time. I'll go ahead and stop talking for the first cycle part because we know that. Okay, so there's that NADH being charged, and another one being charged, ATP being charged, FADH2, and NADH. And as I said, the key is to actually have as many. So again, the purpose of the Krebs cycle is to charge those half batteries, those chemicals that act like batteries, which are NAD and FAD, into fully charged batteries, being NADH and FADH2. Now for the next cycle, we're going to use, or not next stage, we're going to use all those NADHs and FADH2s to actually produce ATP. Okay, so let's take a quick overview of what we already know. And the first step, which is furthest to the left, is glycolysis. We know that's where glucose is in the cytoplasm, and with the help of 2 ATPs, it gets broken down, and 2 ATPs are produced from ADP, and 2 NADHs are made from NADH, and that transforms it to a pyruvate. Then the next stage is, and that's still in that sort of greenish box on the left, the next stage you'll see the arrow where it's moving into the mitochondria, and that pyruvate, through the help of enzymes, has been changed its structure to be acetyl-CoA. What we don't see here, but is in fact the case, is in that whole process 2 NADHs are made. You don't need to worry about it, just know that NADH is also made in that stage. Then we have the pink box, which is the citric acid cycle. That's the same thing as the Krebs cycle, and remember, that's the cycle where we want to charge those NADHs and FADH2s, and you still have 2 ATPs made. The last stage is that blue box to the far right, which is oxidative phosphorylation, and that's more or less the electron transport chain. So we're going to play this and mute it and have it move along. Just keep in mind that for glycolysis, you need to know that 2 ATPs are made along with 2 NADHs. For the Krebs cycle, you need to know that 6 FADHs are made and 2 NADHs, 6 NADHs, 2 FADH2s, and 2 ATPs. So far we have 4 ATPs. And here's that electron transport chain. So this is happening in the membrane of the thylakoid. You can look above on that little box to see exactly where that is. And that NADH is going to attach just briefly onto the protein, drops its electrons off, and as these electrons are in that protein, 2 hydrogens move through. You have these electrons being transferred from one to the next, and with each of those larger proteins, hydrogen moves through until there's this oxygen, that red ball waiting there. That oxygen is going to pick up the 2 electrons, and now it's a charged oxygen, and 2 hydrogens are going to attach to it. So you notice in that orange area, and look above in that right-hand corner, that dark orange area has a lot of hydrogen in it right now. Because those electrons pump that hydrogen in there. And on the far right, you have your ATP synthase. So we know that hydrogen moves from an area of high concentration to an area of low concentration, and those hydrogens are going to move right through. And as they move through, the ADP gets transformed into ATP, having now made ATP, much more ATP. And you notice how many ATPs this is making compared to the other processes. The other step is that FADH2 attaches onto the second of those proteins, and so it also dumps its electrons, so that you have actually 2 chemicals dumping their electrons, both the NADH and the FADH2, to make